crystallization from UFDF to harvest. UFDF, if you're not familiar, is ultrafiltration / diafiltration, or more colloquially known as TFF in most cases. And then harvest is when we pull the crystals out. So there's actually another stage in what we do. Our technology, Crystalomics, is really a formulation technology that comes after the crystallization stage. So we'll reformulate the crystals so that they actually can be injected into a human and used as a biotherapeutic. So today we'll be talking about a background on Althea itself and my group, Crystalomics at Althea. We'll talk about a typical protein crystallization process workflow, and then we'll look at a switch from the typical process to a single vessel process with process analytical technology. And then we'll look at 2 case studies. And we'll look at how an operator can actually use this technology to control crystallization and actually change how the crystallization is working mid-process. So at Althea, we're largely a contract manufacturing organization. We make drug substance in our biologics group, which could be a protein or a plasmid. We do drug product fill finish and complex formulations for soluble drugs of those sorts. And then we also do formulation of protein crystals for injections as well. And then we have a new antibody drug conjugate facility that is in construction right now, but they have capabilities to run process development and analytical methodologies right now. So you can see we also provide services for preclinical and all phases of clinical trials, as well as commercial developments. A couple of years ago, this wasn't true. We've recently moved into the commercial space, so that's an exciting venture for us. However, our technology is before any of these clinical phases right now. So if you also look at Althea overall, you can see that we do analytical method development. We help you make standards if that's what you need. We do stability and release of lots manufactured at our facilities. And then we also do comparability studies. If you look at the manufacturing size, we can take you through process development all the way to fill finish. We can do lyo, complex formulations, and production. So what is Crystalomics? Like I said before, it's ultimately a formulation technology. Our goal is to take a first-generation drug and develop a new formulation for it to make it into a second-generation drug to extend the product lifetime. Our end goal is to really get high protein concentrations in our formulations. We've seen some cases where we can get over 350 mg per mil, which is quite incredible if you look at a standard monoclonal antibody dose. We have several top pharma companies that we're working with right now to do this work. Since we don't have our own drug pipelines, we don't have a discovery group, so that's key to our process and our business model as a whole. We also have the ability to conjugate protein crystals with polymeric amino acids to extend their release profile and make extended release drugs. I'll be talking about HGH today, which ultimately is one of these drugs. We conjugate it with polyarginine and then reformulate it into its ultimate formulation vessel or vehicle so that it can be injected. This is actually a syringe of clinical phase 2 HGH. So then we also target, mostly in our formulation technology, monoclonal antibodies. And the main idea behind that is to convert intravenous injection into subcutaneous. And that's by reducing the overall volume that we're injecting. And hopefully that will help us transform daily to weekly injections and help patients stay more comfortable in their treatment process. So if you come to us with a new molecule, this is the general workflow that we go through in Crystalomics. We'll get a raw material from you, the client, and we'll ask for certain analytical packages. If you don't have them, we can develop them, but it's much easier if you've already developed this drug and methodologies to measure its activity or different forms of aggregation it may be prone to, different chemical changes that it may happen. It's good to get those up front because then we can do our process and then analyze it on the spot and determine during the process development stage if our process is actually changing your drug or if it's staying in its original form, which is very important for monoclonal antibodies specifically. We do an initial biophysical characterization where we also determine what buffer the protein might be preferred to be in to do a good crystallization. This is very important because you want a monomeric species, so you use things like DLS to help us do this. We'll take your raw material then and we'll do a sparse crystallization screen. We have a high throughput system right now that is capable of running around 5,000 screening conditions per molecule, and that's our standard. So we can run that for you and analyze the results, pick a few front runners, and decide on what the lead condition is, and then we'll go and optimize it from there. And we'll do a kind of recursive process development stage. At that stage, it'll take you from microliters up to mils, and so on and so forth. And then we'll eventually start doing some formulation work and start trying to figure out what formulation your crystals may be most compatible with. And then eventually we'll get to the stage that I'm talking about today, which is the bench scale process development, and then also monitoring of this, and that's where scale up and PAT come into play. And then eventually we'll move into large scale manufacturing, and we'll develop a tech transfer package that can go into our GMP suites, and we'll make your product in a clean room, complying with CGMP standards. So to go over the batch crystallization process, we'll look at the typical two-vessel approach. So we start here with our purified HGH. This came from a protein purification process. It may not be in the buffer that we've deemed acceptable to start the crystallization process. So initially we'll filter it and then go into a UFDF vessel. And then the first stage is most likely concentration to improve the efficiency of our UFDF overall. And we'll do tangential flow filtration after we reach our concentration and add in our diafiltration buffer. Typically do 5 to 7 buffer exchanges to get it into what we want. And then it'll be ready to go off to crystallization. So at that stage, we would filter it again. And then it'll go into our crystallization vessel, which is temperature controlled and may feature some process analytical technology depending on how we've developed the process. And we'll add in the crystallization buffer excipients and monitor the crystallization. And then at this stage is where we would add in the second formulation stage. And we'd actually set up a hollow fiber TFF over here and do another TFF process. But this time, the TFF will actually work on a solid suspension of particles. So at that stage, we're really exchanging the solvent that's around the crystals, but also possibly solvent within the pores of the crystals themselves, which is a non-trivial task because if you pull or change out the solvent inside your crystal lattice itself, it is in danger of collapsing or changing the conformation of the protein or the crystals themselves. So then to move on to what I did in these experiments is I moved to a single vessel process. This time we'll take our purified HGH, we'll filter it straight into a single vessel that will perform the initial TFF in, and we'll do kind of the same thing where we'll concentrate it to the concentration that may be most efficient for the diafiltration process. We'll start adding in our diafiltration buffer, and then eventually once we reach completion, we'll just directly add our crystallization buffer and then start the crystallization process. And once that is completed, you could do the same thing where you put your hollow fiber on into the place of your original TFF cartridge and do the secondary buffer exchange to formulate your crystals all in the same vessel. So this gives a couple of advantages, especially if you put in some process analytical technology here. You can monitor all stages with the same probes, do kind of one cycle for everything. So some of the advantages more specifically is we reduce the number of CIP steps, which will help us in validation as well as the actual execution stages. We'll reduce the number of filtration steps, which kind of has the same effect on what we're trying to do. We'll reduce the amount of validation and the time it takes to kind of do everything. And we'll reduce the overall space needed, which may allow us to move the process from one of our larger manufacturing suites to a smaller one, which has our own operational advantages. And then we can also do what I was saying where we add in a hollow fiber into the same basic setup and then kind of do everything in one pot. So the first case study I have here is 100 mil crystallization in the EasyMax reactor. So in this setup, we're doing the TFF in the EasyMax itself. You can see over here we have our diafiltration buffer. This is after the initial concentration stage. And we're slowly adding our diafiltrate into here. We have a scale with rigging up into a computer over here on the side that will show us what the permeate outflow is so that we can match the inflow with the outflow and have a good, nice, efficient diafiltration. And then we'll start the crystallization after this. So a little details about our setup. We have a Pelican 5 kilodalton molecular weight cutoff TFF here. Our protein of interest, HCH, is 22 kilodaltons. Our volume of starting material is 400 mils. So you can see the concentration step in this case is necessary to even fit all of our material in the vessel itself. We start around 9 mg per mil, and our final concentration is targeting 15 mg per mil, but we need to concentrate upwards of 30 mg per mil to leave room for all of our excipients. So what I was trying to show you guys here is what an operator may see while they're doing the actual crystallization. If we look at the top half, we have our trends over time. Right now, this is a snapshot at the end of crystallization. So in real time, this left-hand axis might be constantly changing and auto-scaling. So right now, you can see in this process, once we hit these peaks, at least for these green and red lines, our auto-scaling would really be fixed since everything goes down after that. And then our fines kind of continues upwards, but not out of control. It's important to highlight that we have 3 different scales here. Our fine scale is on a much larger order, well, I guess 1 order of magnitude larger than our 10 to 30 bin, and then our 30 and above bin is on another order of magnitude smaller. Basically how this was working, we were monitoring the reaction throughout the process. So we don't really expect to see any a large number of particles itself. So that kind of confirmed that our TFF was working how we wanted it to. This crystallization itself uses a shear-induced nucleation stage. So when we're ready to start the crystallization, we would ramp up the RPM to 250, and then we'd start adding our excipients. We also know in this case, one of our excipients catalyzes the reaction more than the other 2. So we add in the non-catalytic excipients first, and then right once we add in the, in this case, sodium acetate, we start a timer for 5 minutes, where after the 5 minutes, we'll reduce the RPM back to 100 to allow for a good growth phase. At this point, we see an immediate skyrocketing of the larger species in our system. This is due to basically an immediate amorphous globular precipitation phase that's occurring. But at the same time, we start getting the growth of our nuclei that were created in that first 5 minutes. So over here on the bottom half, we see our particle size distributions for everything. To give you a basis for everything, the dotted lines are the count-based, and the solid lines are squared weight distributions. And over here on the right-hand side, you can see the critical time points that I've chosen for this experiment. 5 minutes and 3 seconds, the first time point, is right before we add the excipients. So that's showing you a nice flat line beforehand. We're not seeing very many counts across the board. At the second time point, we see our median at around 20 microns, and that corresponds to the very max of this initial rise. And around 7 minutes, kind of after this next inflection point, we see our particle size distribution continue to move to the left. That's attributed to the 30 to 100 micron particles both breaking up and redissolving into solution, and then feeding those original nuclei, which is where you see some of this growth. But then it also feeds into this green line going down slowly, and eventually it levels off because this green line also feeds into the fines by breaking it apart and also redissolving to help grow on the small particles. And then when you continue to go down here to this 13-hour time point, we continue to see our particle size distribution move to our left. But between our 13-hour time point and our 20-hour time point at harvest, It doesn't appear to have much of a change, but we know in this part of the process, if we cap the reaction around 13 hours, the particles may not be as robust and they might not be as sturdy, which is detrimental to the downstream handling after this step. So we continue to let this process run until we can take offline measurements and determine that we're at a good place to continue and move on to the next stage. So when we're doing this analysis in the small scale, before we move up to a scale that we can use the FBRM probe, our main source of particle size distribution is a FlowCAM, which is an image analysis tool. So this has a small flow cell that you push your particles through, and you measure each individual picture itself. So you get a very large number of literally pictures of your crystals, and then you also get these different distributions. So in the FlowCAM, a heavy emphasis is placed on counts, but they also transform your 2D images into three-dimensional spherical particles and try to determine what the estimated spherical diameter would be for those systems. This is very similar to the square weighting versus no weighting counts over here on the FBRM. I guess this is also an important time to say that we don't really expect these numbers to line up exactly because the measurements are made in fundamentally different ways. However, we would like to compare them, and we would like to try to translate the numbers between the two so that once we get into the tank, we can kind of reduce the number of offline measurements we need to take with the FlowCAM, and that's kind of why the FBRM is really important to us. So when we look over here, we can see that the estimated spherical diameter average for count space is 1.4 microns, which is pretty small. Our target particle size distribution is basically everything less than 10 microns to achieve high concentration, low viscosity injectables. That's what works best through a 27 gauge needle. And then you can see here that when you actually start looking at the bins that we've generated that we're monitoring in the FBRM, 99.98 are in this less than 10 micron range. This is also an important time to say that when you use the FlowCAM, you don't just take your full slurry and put it into this flow cell. You have to actually dilute the slurry to a concentration that will provide enough separation between the particles to capture individual frames and not kind of group particles together and give you a estimated size that's much larger than what they actually are. So in this left-hand size here, we're doing an offline measurement, and we're also augmenting our sample to measure it. So then on the right-hand side here, when we're using our FBRM online, this is directly in our tank. We're looking at this real time. This is a full slurry. We're not changing our molecule. However, we're measuring with several algorithms a particle size distribution and not measuring each particle individually. And this gives rise to these differences in the size of the particles when you look at the particle size distribution. So you can tell here, these are larger values than on the left. And when you start looking at the bins, we start seeing this 10 to 30 group is rising to above 10%, and you're even getting some greater than 30 micron particles that you didn't see in the FlowCAM. So that was kind of a baseline for us. And we looked at that and said, okay, so now we have an idea of what we're targeting in terms of FBRM. This is what we've done in the small scale. We know we get this every time when we do this optimized process, and this is what we're getting now at our harvest. So this is kind of our target endpoint that we want to use whenever we're manufacturing HGH in this optimized process. So then if we go back to our other more traditional route of analysis in small scale crystallization, the compound microscope is kind of our best friend. So what you can see here in the different sizes is that at 10x, we get a nice even scattering of these particles across the whole slide. If there's significant agglomeration, you would see less of this sand pattern and more of single points that may have larger birefringence or other properties that would kind of make them, it would be less advantageous to the whole system. And then here at 100x, you can really start to see the definition of the particles because they're so small. So then if we go to my second case study, which is a 2.5 liter crystallization, this is kind of not a worst case scenario, but a crystallization gone wrong. And in this experiment, we see some things that at first we didn't understand, and then closer to the end of the experiment, we try to rein in the crystallization and see if we could get the same outcome, even though the beginning of the experiment was not as tuned to our specifications. So here we have our second setup. You can see that our EasyMax was replaced by a non-jacketed bioreactor. Our first process was performed at 33C, but we have data at 22C showing that our crystallization can still take place in a longer period of time, but kind of achieve the same particle size distribution after 8 or more hours longer than the original. Once again, we have a 2-liter vessel, but we start with double the starting material. So we do the same process of concentration beforehand. We're using the same starting material, just much more of it. So we concentrate to the same amount basically before adding our excipients. We've also increased the size of our TFF cartridge to help us provide the same load to flux ratio or load to surface area ratio to achieve the same or similar speeds in the initial diafiltration process. So then here's our second crystallization protocol or profile, and you can see already it looks much different. One of the points I was making before of the auto scaling that happens while you're doing a process, when I finished the TFF process and added the excipients in this experiment, I initially thought everything was kind of going according to plan and going the same way as before, because we saw a nice peak here and then we started seeing a decline of some of our other trends. But after another 2 hours, we saw the massive increase that we saw kind of the first time. So then we started thinking, like, what made this happen? Another thing to note in this crystallization is when you add the sodium acetate, the entire tank turns white due to the amorphous precipitation that you see. So you can't really see what's going on. But as we started to see the decline of this very large particle bin, we started noticing that we actually created a striation in this new vessel. So we determined at that point we had inadequate mixing. And our final time point in this experiment was supposed to be somewhere between 24 and 28 hours. And at that point, when we realized that we reached the same amount of time and we didn't get our nice kind of baselining or flattening of our curves, showing that our process was kind of reaching the terminal phase where it would stay flat for a while and then basically just stabilize, we decided to see if we could reach the same particle size distributions. So to go to the second plot on the bottom here, our first time point, once again, is before we added our excipients. We see a nice flat line. And then our second time point is actually this green particle size distribution, which is much smaller than our other ones. And it's very similar to this 4-hour time point that's at the peak of these growth curves. So that's not, it doesn't look the same as the previous experiment at all. So then we look farther along the line, and then we see this large growth to the right here that slowly moves back to the left. So after this 18-hour time point is when we started thinking about trying to augment our particle size distribution. So the first step we took was to try to increase the RPM of our impeller to see if we could create a larger vortex that would pull in the material at the top of the tank that was striated into the bottom of the tank. What we immediately saw was a big change in our counts, which is largely due to mixing around the FBRM probe itself. So we saw much more fine particles moving around, so we got this original spike. After a certain amount of time, it was approaching the end of the day, and I really wanted to go home and let this run overnight. So instead of letting our RPM stay really high overnight, where we might have a potential situation that with the pump, the overhead stir that we don't use very often, I decided that a recirculation pump might be better. We know from formulating our crystals that once they're at a certain rigidity, that they actually get milled slightly going through a peristaltic pump. So we decided that it might be a good idea to try to both recirculate the top layer back into the bottom layer, but also try to mill those particles as they go through the pump, and that may fix our particle size distribution. So at this stage, I started the recirculation, and when I returned the RPM back down to 100, we can see here that we caused a significant change in our particle size from that small increased shear step. And then after this point, we kind of see the flattening out that we wanted earlier in our reaction. So we sped up the process of our particles getting broken down and growing more fines at the same time. And then when I came back in the morning and wanted to start harvesting the tank, I still could see that there is striation. There is still a top layer and a bottom layer. So then I had to get extra ingenious and trying to figure out a new way to kind of solve this problem, because we wanted to use this material for further formulation development work. So we needed our harvests to be very similar. So what we actually did is we did a 3-step harvest. So our harvest port was very close to the impeller in the bottom layer. So we took out, I think, 500 mils of material at first, which lowered the top level of our reactor down closer to our impeller. And the idea was that if we just lowered the top layer, eventually it'll get mixed into the bottom layer, and then the shear of the impeller will do the work for us. So we let that mix for an hour or so, and then checked we could see from our FBRM that we didn't see a significant particle size distribution change. And that's kind of what we were expecting, because the FBRM was really close to our impeller the whole time. So when we started moving things from the top to the bottom, we saw the same particle size distribution and we didn't see a large increase. Theoretically, we are achieving what we wanted by milling the crystals and putting them back into the same layer. So after enough time, I did the same process again. And then we did it one more time near the end. And we see this small jump in 10 to 30 counts. And that, I'm pretty sure, is an artifact due to our top layer of the solution actually approaching our FBRM probe and then getting some air interfaces in there. But largely, you see this signal stay about the same. And then at this first harvest point is where our last critical time point was. And you can see this curve is much more similar to what we were looking at before in the 100 mil fully optimized batch. So then we can look at what was actually going on in our 2 layers. While this process was going on, FBRM wasn't my only tool. I was also doing offline measurements to kind of verify what I was doing along the way, especially since I knew the top layer was not being measured by the FBRM probe. The FBRM probe was measuring the bottom layer that was having less issues. And you can see from this bottom layer, we're getting a nice even covering across our slide. But when we look at our top layer, we see these agglomerates of crystals that are definitely not in this phase. So basically, what we saw was a significant difference in the growth profiles, and that alerted us to a change. And we were able to use that and think creatively to try to problem solve our way out of this situation. And we also determined that our optimal particle size distribution couldn't be achieved by the crystallization condition itself once it was compromised. However, we could use outside factors to control the particle size distribution at that time. So once again, like before, we were doing offline FlowCAM compared to our online FBRM. And we can see here, before we were looking at a 1.4 count-based mean. Our volume-based mean rose from 8 to 12. And you can see there's slightly more 10 to 30 micron particles. Another kind of artifact from the FlowCAM is when most of your particles are in this very, very small particle size distribution, you see this kind of cubic or quadratic growth from a very low number of particles, because the volume of those particles, if they're 10 times the diameter, they're 1,000 times the volume. So you see this kind of growth out here, and a significant volume percentage is seen in these particles, even though they may not be really visible in the frequency-based counts. But we kind of see the same count-based distributions here. And then when we look over at the FBRM, we're basically right where we were at the end of the 100 mil batch, where our not weighted average is around 5 microns, and then our squared rate is around 13. And then we're seeing less than 10 bin of 86%, and 10 to 30 is around 14, with a very low number of greater than 30 micron particles. So this kind of alerted us that we may have achieved what we wanted to, but we kind of wanted to verify that in doing this multiple stage harvest, we didn't just pull off different layers at different times and kind of augment the way we were measuring our results. So then what I decided to do is employ some statistical analysis. So what the FlowCAM allowed me to do is it gave me individual properties for all of the particles it counted. So I decided to do an ANOVA analysis on both the final bottom layer sample that we saw a picture of before, a sample from the first harvest, a sample from the last harvest, then a sample from the top layer at 43 hours. And what, at the end of the day, we're looking for here is a significant p-value from this test, and 0.0001 is definitely a significant value. So then, since we know that the top layer was significantly different than the other harvest and bottom layers, we went over to our harvest fractions and looked at them comparatively and tried to see if they were statistically significant as well. But as you can see from this p-value, they were not. So our harvest effectively reduced or homogenized our sample enough so that we could theoretically use our different harvest fractions in an ongoing formulation development work to kind of continue our progress, and we didn't have to scrap some of our batch. And then when we compared the 100 mil FBRM at harvest with the 2.5 liter FBRM at harvest, we can see these graphs overlay pretty well on top of each other. The blue run, which is the 100 mil run, had a little bit higher count number. However, due to the different sizes and placement of the FBRM probe in the tank, we're not entirely sure if that number is significant or if that is due to the setup of our system itself, since that has a large influence on the count itself. But at the end of the day, we effectively fixed our crystallization and achieved a similar result to our well-optimized system that we had in place before. So then, in conclusion, the FBRM analysis online can detect deviations from a usual particle size distribution in real time. So that can alert you if your process is starting to fall off course or starting to have issues, which may allow you to save it or may allow you to scrap the run before you've wasted a significant amount of time trying to kind of take care of a crystallization that's already doomed. And then we also determined that if mixing is dramatically different in parts of the vessel, we have to take that into account when we're looking at our FBRM data. And we can't just say, oh, since the probe is in there, it's a gold standard and we're doing fine if it says we're doing fine. You have to also visually look at your tank and make sure it's doing what you want it to be doing. We also discovered that FBRM online analysis agrees well with our offline analysis, although not exactly. And if we characterized our process well before and then do maybe one engineering run with a significant number of samples to verify at each point in the changes in the particle size distribution, we have a similar growth pattern or similar trends between the two measurements. We can use the FBRM probe exclusively instead of trying to do both in further experiments. And then we also determined that separated layers, at least in the case of HGH, can be homogenized with recirculation using a peristaltic pump, as well as changing impeller speeds to kind of assist the breaking up of different particles. In doing this work, this is a great team at Althea and the Crystalomics group, and without them, I wouldn't be able to conduct these experiments. So I'd like to thank them.